It was off.
It was off, but it's still blinking. Oh no, it's off. Okay, thanks. So I go back and I say everything. No. I'm sorry, bad luck. Thanks that you notice it. Back again to the question. How can you predict solvent accessibility? By something other than what we just talked about. You can use hydrophobicity, you can use conservation. Now what I want you to do is develop your own prediction method. Generic prediction method.
You just learn from known solvent accessibility?
Yes, exactly. You can. Know what you put as input. You have tons of data. You have we have 100,000 structures. If you group them, at least 10,000 are sequence unique. So on average you have 200 residues per structure. This is a big data set. So you can do a lot with machine learning. What we use as input. What's wrong? Is it the day? Is it the hour? Okay. Simplest thing is you use the same thing that you used previously for secondary structure prediction. You use profile information from alignments, you use sequence information, you use some additional information about the length of the protein and other things, conservation, and then you have a simple, in this particular case, machine learning device. This in particular case is a neural network, could be anything else. And here I have a particular way of using the output to have a square root function that is the percentage solvent accessibility, so that unit is on whenever it's 0, between 0 and smaller than 1, larger than 0. That is between 1 and 2, 2 and 9, 9 and 16, blah, blah, blah. You immediately see this is by design such that it's more sensitive to more buried. On this end of the buried question is much more sensitive than on that area. And then you can use those predictions and turn it into anything you want. In ASA, you can turn it in relative, you can turn it into two-state model. When you do that, you get accuracy that is in the ballpark of 70-something percent. What we look at is, in fact, this one here. This is secondary structure. This shows you the curves for reliability index. So when you predict for everything, the two-state accuracy is about 74% here. When you look at the 40% most strongly predicted residues, then the two-state accuracy for those is sort of 86%. And I'm afraid I cannot remember whether this is a two-state. Yes, it must be a two-state number. So this one here is secondary structure in black. The dashed line is solvent accessibility. Secondary structure here is a three-state model. Solvent accessibility is a two-state model. The random levels are 35 versus sort of 50-something. Nevertheless, your prediction method reaches the same level. How could that be? Let's put it differently. So we get overall, I cannot remember, a level of accuracy that is about 75%. But what we observe is we cannot train much higher than 80. No matter what I do to this neural network, I blow it up in terms of the parameter space to something that is beyond reason, and I still cannot learn it. Why? Why may that be the case? When you blow up a machine learning device, what could be in the way of learning the training set? Well, essentially what you learn in the training set is to classify the input vector into some output. Say in the two-state model, buried or exposed. If you had two vectors, one you're supposed to train as buried, and the other one is a very similar or identical vector, and you're supposed to say exposed. Then that's a conflict. How could that conflict arise? How could it happen to you that you have two possibly even identical input vectors, or very similar ones, yet one is classified as buried and the other one is exposed?
Don't we want that?
Hmm?
Don't we want that? So that
No, it's the same vector, it's the same input vector that has two different opposing classifications. So at one training instant, the system is trained to learn this vector is buried, and another training instant, the same vector is exposed. Obviously, this is a tough task, impossible task, to be more precise. How could that happen? What could generate such a situation?
you overtrain.
No, we do overtrain. We do train beyond overtrain. Still cannot learn it. We cannot learn it because we then observe that there are identical vectors that are mapped to two different states. How could that be the case?
So one classification in the training.
Experimental error is one possible reason. There's another reason. It's correct. That's one possible reason. Another possible reason is that the input vector that you have is not completely classifying the problem. For instance, you may remember in the context of secondary structure prediction, I told you you could put 11 consecutive residues. You can find an 11-mer, a pentapeptide of 11 residues, that in one part of the protein adopts an alpha helix, and the same 11-mer, and another part of the protein adopts a beta strand. Right? So if a machine learning device saw only the sequence, then they would get the same vector at two different points and would have to train two different things. In that particular context, I told you that using the evolutionary information helps you to get out of that dilemma. And through the evolutionary information, you could actually correctly predict these two positions. But ultimately, what the example tells you is that if my problem is not well defined from the input, I cannot solve it. And that may be the case here. Now, this relates to something else. Remember when I say that in the Q2, the performance is 75%. And when I ask you, is that good or bad? There are 2 points that you have to ask for. What are they? To put this into perspective, any number like that. Any prediction method, you have to assess against what's the worst you can do and what's the best you can do. What's the worst you can do? It's a two-state system. So they're equally distributed. I don't know what's happening today. Okay, maybe I don't entirely. So let me try some new approach here. From now on, for today, whatever explains the situation, from now on, I'm just going to go ahead and do my thing and not ask anymore. Is that the right conclusion? One. I want you to shake. I want a clear opinion on this. This is a yes or no. There is no abstain from this one. And I have one opinion.
I'm a no. But I'm a no for.
I know. We have a 50-50 tie. Well, the number is such that there will be not a tie at the end. And you better answer quickly, because otherwise you're going to be the one who's going to be damned because you're the tiebreaker. The last one may be the other. Half of the room will possibly not like the other one, so answer quicker. No, yes?
Yes.
No.
I told you. August, you waited too long. Bad luck.
Sorry again. If I say no now,
then 2 will not like you. And if you say yes, the other 2 will not like you. That is true. In that sense, it's the same. And you have no memory who were the 2, so it may be the same for you.
What was the question? You're going to answer questions?
That is a very, very helpful trick to pretend that he is less guilty. Socially, you did the right thing. I stop asking questions from now on and do my thing. Yes or no?
I'm for questions.
Okay.
If it's not too
That's a no. Okay, so tell me whether they're too annoying. I mean, at least stop. Anyway, so we have a distribution that is 50% of the data is buried and 50% of the data is exposed. What's the random probability of getting a two-state prediction right? 50%, right? Good. So the bottom, the worst you can do is 50%. Now the question is, what's the top you can do? Okay, I don't want a number from you. What I want from you is how could you assess what the top is? Okay, you could Google, you could ask somebody who knows. Say you were really the first. When I did this, there were all kinds of answers, but actually nobody really had thought this through. So you have to think it through yourself. How would you do that? What could you do?
For a binary classifier, you can just vary the threshold for how you classify it and then plot.
That would possibly change your random, and that would possibly change this number. But it would not tell you that is the Q2 of the worst. That's the Q2U. And now what I want to have is the Q2 best. Again, you may argue Q2 best is 100%. But I want to argue this is not the case. It will be a number smaller than 100%. What could you do to establish what is the best possible? This is not another method. Again, you will compare yourself against other methods. That's not the point. I'm talking about a different space that defines more the background of your problem. If you were the first method, you still would have to do what's random and what is possibly best. Give me ideas how you can assess or get at a number for what is possibly best. There are 3 or 4 different things.
Wrong with your method, change what.
No. That is just, I'm talking about, in some sense I'm talking about before you develop a method. Before you sit down your first foot and say I have 75. I want you to already have an expectation whether that is going to be high or not. And one level that you can have the expectation for is random. Now give me another. Well, another one could be that you have hydrophobicity in here, which in some sense would be, or conservation, those would be other methods. That's not what I mean. I mean a top method. So something that is a ceiling, that is, you cannot do better than that. How could you estimate that? Any idea?
You look at your data set and see
Yeah.
How different it is, how large it is, so how much redundancy you have in it.
What would that tell you? That would give you, in some sense, that would give you a standard deviation for the number of buried residues, or what? Or would give you a standard deviation for your performance. That would give you a standard deviation on this plus minus here, and you have to absolutely do that. Highly appreciated comment. But that is not giving you the ceiling. That clearly is part of what you have to do in order to assess your method. Is it that window or?
No, it's fine.
So, one point is experimental inaccuracy. How reliable is the assignment from experiments to this number? How can you check that? No. Did somebody fool me? There was somebody knocking, right? So, thank you. I had for a minute, I had one.
You count your classifications from the same input vector which can't classify?
That would be one way. In fact, you essentially go back to that number. So you ask how many of the cases are such that I have a double assignment. That is true. And that is a perfect solution. And not a perfect, it's one possible solution, and that would in fact do it. In that sense, however, the problem would be that still this number would be biased and some might be biased, by the way you design your input. I'm looking, you see, random is not designed by your method, or it has nothing to do with your method. Before you ever develop, random is there. I'm trying to see something here before you develop the method. And so one way of doing it is you simply take two proteins of the same sequence for which you have two different structures. These days we have a lot of these examples. We have a lot of structures that are done again because of a variety of reasons. And we simply compare them in terms of their solvent accessibility assignment and ask, we just take one as a prediction method for the other one. Of course, it's nonsense. But this is one way in which I can define a ceiling. And that will most likely not be 100%. Another way of doing it is I take proteins that have similar structures in 3D. And I see, when I look at two proteins and I classify by CAF or SCOP, I classify them as similar structures. What is their similarity in terms of solvent accessibility? So again, I will not reach that with a prediction method. This means that two proteins are so similar in their three-dimensional structure that I can recognize the three-dimensional object is the same. Most likely, their similarity in solvent accessibility will be higher than what I can achieve. That's the second ceiling. And the second one would already, you expect to be lower than the first. Now comes the third one. The third one is simply, and that typically is more important usually when you do this kind of thing. What are you up against? So, what I mean here is we are trying to predict 1D structure. 1D structure is of any use only if you don't have 3D structure information. If you do not have comparative modeling, or from comparative modeling you get 3D information. So when you don't have a comparative model, then you want whatever you can get. And 1D may be the only thing you can get, and that may be satisfactory for you. That means that in an example where you can do comparative modeling, your method will not be applied. So in that sense, you could look at comparative modeling. You could ask, what's the average accuracy of comparative modeling to predict solvent accessibility? Clearly, your method will not compete with comparative modeling because you're trying to carve out a space where comparative modeling cannot reach. Okay? So that's another way of defining a ceiling. That is the part that I wanted to make, and you will find when you do that, you will find that in fact comparative modeling can not reach these levels. So there is some problem in the way we define the input. Possibly it's the problem in the way you define the problem. Proteins, so solvent accessibility just can easily move around because there is some mobility. So getting it a little bit out, a little bit higher changes this value quite dramatically. That's why it's not conserved between homologs, or not that well conserved as secondary structure. Is the statement here, and somehow you can see it. But I'm going to go over that, and I'm just going to go quickly through a few examples here. In this particular example, that show how you can use solvent accessibility, that's conserved from the group of Nirbental in Tel Aviv, showing a 3D structure, overlaying over the 3D structure conservation. Very dark red is high conservation, very blue is low conservation. In some sense, this is a counterexample of, or is the example that you showed before. So if you saw that, you somehow, Virginia, you had that, what would you say this one is? Yeah, and that's exactly what it is. So this is conserved as a remarkable way from structures, or putting alignment information onto structures and immediately seeing binding sites. And sometimes it's amazing what you can see with that. Here's an example where you see a pocket inside through this, and you can use that also to somehow derive some features of proteins and function. Now you could imagine that you use predictions of solvent accessibility to identify domains. And now I'm going to present you something that somehow works, but there's another aspect to it that I find remarkable. That's the main reason why I present it. It does not quite work anymore for defining domains. So the idea is the following. You look at a protein domain, you look at a fragment thereof, you look at a chain or a fragment thereof, and for the fragments you always see that the fragments somehow have fewer contact density. Fewer contact density, if you predict contacts, you can get at that. How can you use solvent accessibility to get at that sort of feature? don't have predictions of contacts, so you immediately see from these images the idea that domains are compact units. In fact, that's the way you can assign domains from structures. If I gave you a structure and I did not give you scarf or scop, you had never seen a domain before, so you cannot 3D align and find other domains. But instead, the task would be to identify a domain from one three-dimensional structure. Then you would simply, one way would be to look for the densest region. For something where the number of contacts inside is higher than the contact between this and something else, right? And you see that the contact density here is lower than here. But we don't have contact density, we have solvent accessibility. What could you look at to sort of approximate that idea? Well, that's, yeah?
I think this couldn't work if you look at per residue solvent accessibility.
Yes, then?
Because you can't see a difference.
That's exactly what I'm going to challenge. So let's begin with the following. What I show here is the length of a protein. Okay? Now, assume every amino acid would be a cube. And the protein would be made up of cubes. The whole protein would be a cube made up of small cubes. If I do that, I can clearly relate the length, the number of cubes, to the number of cubes I will have on the surface. It's a very, very simple function shown here. I can refine my model. Instead of cubes now, I have spheres. When I take spheres, okay, you cannot do that before you leave the room, but if you find your way to the ESA, by the time you reach the ESA, you can work out the math to do this curve. Or you Google. That's this curve. Everything is sphere, all amino acids are the same length. Just the number of spheres will influence the number of spheres on the surface. Why is it such a shape? Essentially it's a square root function, right? It's clear. It's the surface, it's the volume of a cube with respect to the surface. The growth, and that in fact is exactly why water drops look the way they look. Blue are proteins. That's remarkable, huh? It's remarkable that proteins look, the model of every amino acid being the same size and proteins essentially being spheres is remarkably approximating the observation of length, number of residues, versus solvent accessibility. Exposed residues, surface residues is what I show in blue. Isn't that remarkable? You find not? Okay, so this for me, this is the one of the few slides that has survived history. That was simply enough, where something that you can work out in your head really relates to something you observe. Most often proteins are way more complicated than this. But now, since you have this function, and you could take that or you could put a fit into the blue.
I have a question. Why does it say proteins are amazingly cubic? Isn't the idea that they are not spheric? So
Yeah.
It's very common, then it's better.
The idea that you build a protein from a cube and that every amino acid is a cube is a pretty dumb idea, okay? And that we get anything near the blue, for me, was very surprising.
Okay.
Cubic is a misspell, and it's actually an old play on a theme that I want to. There's something to do with Columbia University, but still, it is amazing to me that the cube gets you anywhere near result.
But the sphere does get you that.
Yeah. But again, amazingly spherical. Yes. And then actually, if you can actually top this by making the over, so you still. Let's not get into that. So you can actually get surprisingly closer by assuming that all amino acids are spheres. No, I'm the kid again that I was when I did this. So still the amino acids are spheres, but the protein no longer is a sphere, but it actually is sort of a little bit elongated, which in fact it is elliptical. And then you get really down here. So you get close to a fit of you got close to a fit of the data those days. Anyway, now back to that story. You can predict the number of residues exposed, you know the number of residues, and you know exactly where you expect to be, and you know whether you're deviating from it. So you immediately see where they are clearly not compact. And in fact, you can even introduce cuts to optimize the compactness of all of these without ever predicting contact. And it worked in the past, it doesn't work anymore in the current, because in the past we just had fewer examples, and for the space of sequence, the complexity of the sequence space 20 years ago, it worked. Today, there are too many outliers and too many other issues. But it's remarkably successful. Let's get back into 10 minutes membrane protein wrap up. But where did this? I believe I just had a point where I said, do you remember where we, this is only 5 weeks ago, right? Or, so you must remember where we stopped. No. Did we talk about membrane prediction, the TM using hidden Markov models for membrane protein prediction? So again, there are two, I believe, quickly, yes, we talked about it because you were in the room, and that was the Hungarian slide.
I think we talked about the evaluation Jonas did.
Jonas presented in this group?
No, but you talked about it.
Oh, so I showed you that that this was all wrong, and that we have to redo the. We got it somewhere here. Not entirely sure. So the random. I talked about the assignment method, exactly the true positives, and we talked about the segment idea. That we have to measure somehow how well we get segments, and the segment idea is that we're from now on going to look at segment overlap between the observed transmembrane helix and the predicted membrane helix. We allow them to differ by at most 5 residues on either side. And if they differ by less than 5 on the sides, then we say that the membrane helix is correctly predicted. And then I define a value that is called Q or K, which means, right, I found it. That was the hint for myself. So that value measures how often, for how many proteins, which fraction of the proteins do I predict all membrane helices right. Again, right is defined as I'm within 5 on the ends, and I get the number of helices right. Okay?
I have a question, because I think we talked about that last time, why it's 5 residues. So we discussed it.
That's the answer. Very often, that's the answer. Sometimes people don't admit it to it, but there is absolutely no other answer. We did check a variety of values. So let's go backwards. When the number is too high, then essentially that means that you begin to call an overlap of this type. This is observed, this is predicted. So you allow, in this particular case, maybe visually what I draw is 14 difference. And then you begin to call things that are really not related and really incorrect predictions to be right. Historically, this is what people, including I, had done. And that was very much driven by low-resolution experiments. In low-resolution experiments, this turned out to be good enough to reproduce the experiment. Let's not get into the details why that is so. But there are good reasons why this is not the right way of doing it. So on the one extreme, we know that the number of overlap must be larger than something. So now we get into the issue 5, 6, 7, 8, 3, 4, 5. And we looked at a variety of features and couldn't distinguish and ended up with 5. And I suspect that 4 or 6 would have been equally good, or 7. And that ultimately there was a preference looking at 5 because our eyes are focused on 5 because we have 5 figures. I ultimately believe very often in science you have some dumb decision like that. Ultimately because it's as good as any other by the criteria we had looked at. We did go about it in a sort of semi-systematic way, but not in a fully systematic way, okay? Fully systematic would be to really one way in which you end. Is that enough? Bunch of methods, different levels of sort of random. This really is totally random. That's semi-random. That means that I know the number of helices, I don't know where to place them. That is just the simplest hydrophobicity index. Typically on this side here you have methods. TopPred is the one that I showed you first from Gunnar van Heenje, that predicts in an advanced way hydrophobicity and puts membrane helices using the positive inside rule. Was developed over 20 years ago. That's where it reaches. Those here are much newer methods that essentially you can think about them as 20 years of development. And those are the levels we get. So again, let's look at the numbers first. About the seeding here is about 60% of the proteins are predicted completely correct by the plus minus 5. If we change this number plus minus 5, this number goes. So if we reduce it, it must be they are allowed to differ by only 4, and it slowly gets down. If we increase it, they slowly get up. And there was a saturation issue where things did not change that much, and I believe 5 was somehow a good compromise. Anyway, back to the point. 60 is sort of ceiling. If you want 30 here, thin is the lowest, but you may also argue 20 is the lowest, or you may in fact argue that the simplest, dumbest hydrophobicity reaches sort of 40-something%, and that in fact is the lowest, because anybody of you could implement that within 5 minutes, or 10 minutes, or 20 minutes, or whatever it takes. But this is not worth any time investing. So by that logic, that clearly is one way to define the lowest, the worst possible. And you see a variety of things. You see that some advanced methods are not much better than that. You see that the sort of older methods are slightly worse than the newer method, but not really by a standard deviation. You see that there are now new methods that really seem to be doing better than the pack, even by the standard deviation here, the standard errors. So this blue one, polyphobius, in that data set on these 95 proteins does better. But now we get into new ways of looking at it. So this should read 22 is a technical issue, and this should read 73. 22 plus 73 is 95. We have old and new. What that means here is those are proteins that could have been used by some of the more modern methods for training. So those were crystal structures solved over the last 15 or 20 years. And you see that for this set here, the older methods are sort of in some sense more different than for the newer set. These are proteins that are new to all of these methods. All of these are new to the methods that are on this side. You see that there's a difference between these and these. So the ones that have been used before perform much better than the ones that are used here. What could the reason for that be? That's one possible reason, yes, totally. So that we added new proteins that are completely different from what we had seen before. Any other reason you could imagine?
The new methods also take some kind of information which came from the old data sets.
Yes. Yes. So they are in fact largely trained on the old data set. That's why she says, yes, they were trained on these old proteins, but the new proteins look different. They were not trained on them, they look different. That's why they don't get them. So you're not saying anything else, do you?
But we have some new methods which
Yeah.
are also trained in the old data.
Yeah.
Okay.
So the new methods are essentially the way it is. I'm sorry, I'm not very clear because I'm rushing through something that it was supposed to be an entire hour in 5 minutes. Most of these methods were trained on most of these proteins, while these methods trained 20 years ago used, in fact, and I'm the author for one of these here, I had 4 proteins of these 95. So most of these proteins I had not used. Gunnar had in fact none of these. Yes, he also had 4. Actually, had the same number. But these 2 here have a very, HMM top had a little bit more, but not much more. So most of these here, in fact, did not use these new ones, but they, in fact, mostly used the 73. But again, so the typical explanation when you see something like old, new, is overtraining. I don't know, I'm just, making up a completely nonsense number. In 90.999% of the cases, that is the right explanation, overtraining. I don't know why you picked the 1 in a 1,000 exception explanation. That is in fact the case. Do you have a reason for that? In this particular case, it is true. You were right, Virginia. But why? Why did you pick that?
Isn't it kind of the same thing? Because when the first methods didn't have a large data set, so it could be more different.
Yes.
And the new added ones were more similar to
Exactly.
the old data set, so they overtrained.
That's exactly the point. So they are lower here, although they used almost the entire set here has not been used for training in these 2, or these 4 here, and the same is true here. Nevertheless, these 4 clearly are lower for that. And that difference here, between wherever you put the number here, 50 and 30, that difference is their new features and new structures. And some of these features are, when these methods were developed, or most of these proteins are such that the helix really sits orthogonal in the membrane. Many of these are like that and have re-entry helices. So the helix going in and out. And those are kind of features that are very tough to handle. In some sense, you see, since the newer methods here on most of the data are higher than the older ones, while the difference here is much smaller, you see that there is some overtraining effect. So in some sense, the 20% jump you explain by novelty. But if you looked at some of these here, you have a sort of 35% jump. And what comes on top of the 20 most likely is overtraining. So you see 2 different effects. Okay? And that's a unique situation. It's not often the case that you see this so clearly. Let me get into something else. That's an important point. What this one here shows is the distribution of things where, for all the methods and all the proteins, 95 proteins, you count the number of methods, I just say 10, I can't remember what the number is. So you have 950 cases as a distribution, and you ask for one particular case of the prediction of the 900, what's the distribution of those where the number of helices is exactly right? That's the peak. You predict 1 helix too many, and 1 helix too few. Those are the peaks. And you see that plus minus 1 helix, 0 or plus minus 1 helix, carries almost the entire distribution. Right? That means that in some sense, prediction methods are already very good. They get within 1 membrane helix in the vast majority of all cases, all methods. That's remarkable, right? But here's a different sort of way of looking at it. We can, on the one hand, that's another way to say it again, it's good. What I'm asking here is, I give the method a bunch of membrane proteins. Does it recognize them as membrane proteins? Most of the cases, yes, is the answer. Now I give a bunch of non-membrane proteins, and I ask, are they correctly characterized as non-membrane proteins. And there you see that some methods here have error rates that are very low, 1 to 3%. Polyphobius, Phobius, or HMM here is another very good one. And some of the methods, more hydrophobicity based, top pret in particular here, have very high false positive rates. That is because hydrophobicity based methods tend to say, whenever I see hydrophobic region, there's a membrane helix. That is not true, right? Here's another issue, signal peptides. You know what signal peptides are? N-terminus is the beginning of a protein. The N-terminus, you have a region that typically is hydrophobic, is recognized by a protein in order to bring that protein to the surface. All proteins that are secreted, so put out of the cell, most proteins that are secreted have such a signal, and that signal again is recognized by a protein that shuttles you to the membrane. In some cases, the signal peptide is cleaved off. So essentially, the signal peptide can expand to about 50 residues. So you have a 300 residue protein, mind that. 50 residues are cleaved off. And the only purpose of creating those 50 residues is to be cleaved and to be moved. Anyway, in some cases, the signal peptide remains on, is actually hydrophobic, and becomes a membrane helix. This is why many methods confuse signal peptides with transmembrane helices. In fact, the only, so you see the extremely high error rates here, the only methods that sort of get small numbers here, Polyphobius, Philius, are methods that explicitly use the idea of signal peptide. So they predict a signal peptide, a membrane helix, and then they say, if I predict a signal peptide and a membrane helix, I call it signal peptide. But still, we get to relatively high error rates. That is one problem that continues to exist. This is a slide that in detail would say maybe there is some improvement possible, but the background really is that while this image gives the idea that most methods get it mostly right, the prediction of membrane helices continues to be a problem that has to be improved on. Maybe we cannot do that. And maybe we cannot do that because of a variety of reasons that I'm just going to flash by showing you some structures and some problems of structures that I thought that I cannot. It's the wrong part of the talk. I'm sorry. So then, here are some examples. Re-entry helices. You see that this helix here, so you begin here, and that is the loopy part, and then you have actually a helix. The same thing happens here, and these two helices, they actually functionally have one long helix, but they come from two different sides of the molecule. What was this one here? Interface helices. So you have helices that are completely hydrophobic, that are membrane-like helices. They do not go through. They sit on the surface. Now you may argue, okay, I can recognize that because they are too short. Whoops. But that's a problem with the re-entry helices, right? The moment you have suddenly 5 or 8 residual helices, you can no longer say that if you have 5 or 8, they cannot go in, they must be on the surface, because they could be in. In fact, there are more and more classes of re-entry helices and different types. And so these slides are, sorry for moving so much around, from Arne Elofsson. And Arne Elofsson developed a method that predicts re-entry helices. The problem with that method ultimately is that we have very, very small numbers. And then there are many kinds of proteins that have additional problems. So I talked about the angle. I believe the next slide shows that. So we have examples where the membrane helix goes really in strange angles. We have examples where, in this particular case here, where you do have the kind of re-entry helix situation that I showed you before, plus you have a dangling helix here that sits in the membrane. and this is really, really difficult to recognize, this case. Gunnar van Heintje talked about a case where, and this is not shown here, where you had a very, very long helix that was predicted as membrane helix by all methods, and he always saw it in the structure. Then he found some aggregation of this protein as a multimeric aggregation, in which that thing was suddenly inserted in the membrane. So some of these prediction mistakes may not be real mistakes. They may actually be observed in nature. The reason why I say those methods are not good enough is one way in which people, for membrane proteins, I told you, you cannot easily get X-ray structures. Cryo-electron microscopy becomes an issue that is more and more interesting. You can do membrane proteins in cryo-EM. But cryo-EM, you get just the blurb on the surface. What you need to now to do is take the blurb and put the trace of the protein into it. So model it. If you knew where the membrane helices are, you could actually do that. But the problem is that the prediction is too inaccurate to do this at the moment. So that's one of the incentives why we would want to know the membrane helices better, to be more successful in fitting the prediction of membrane helix locations into the images that we see. I leave it there. Thank you for your attention. Sorry for the complication.